Hey everyone, welcome to Gray's Gyan. I'm so glad you're here. Today we're diving into one of the most fundamental topics in cell biology, the nucleus. And when I say fundamental, I mean it literally. Every single thing your body does, every heartbeat, every thought, every breath, is ultimately controlled by what happens inside this tiny structure. Alright, let's get into it. So, what exactly is the nucleus? Let's start with the basics. Look at this diagram on the right. This is figure 1.1 from Gray's Anatomy. It shows a typical animal cell, cut open so we can see everything inside. And right there, that large pink dome-shaped structure in the middle? That's our nucleus. Now, Gray's Anatomy tells us 4 critical things about the nucleus straight away. First, it is generally the largest cell organelle. Intracellular simply means inside the cell. So among all the organelles, the mitochondria, the ribosomes, the endoplasmic reticulum, the nucleus is the biggest. Second, its shape is typically spherical or ellipsoid. Think of it as a sphere or a slightly squashed oval. Its diameter ranges from 3 to 10 micrometers. And fourth, it is the control center of the cell. Everything the cell does is directed from here. Now you might be wondering, why does the size vary between 3 and 10 micrometers? Great question. It depends on the cell type and what that cell is doing. A liver cell that's very metabolically active will have a larger nucleus than a mature red blood cell, which actually has no nucleus at all, interestingly enough. But let's not get ahead of ourselves. The key takeaway here: the nucleus is the largest, most important organelle in the cell. Now here's where it gets really interesting. And this is a topic that confuses a lot of students, so pay close attention. When pathologists and histologists, that is, scientists who study cells and tissues, want to see the nucleus under a microscope, they can't just look at the raw tissue. The cells are transparent. So they use special dyes called stains to make things visible. Gray's Anatomy specifically mentions 2 conventional histological stains. Conventional means they're the standard, routinely used ones. Hematoxylin. This is one of the oldest and most widely used stains in all of medicine. It's extracted from a Central American tree called Hematoxylum campuchianum. Under the microscope, it stains the nucleus a deep blue-purple color. Toluidine blue. This is another basic dye that also stains DNA and RNA intensely blue. It's particularly useful in electron microscopy preparation and in staining nerve tissue. Now, and this is the critical bit, why do these stains color the nucleus? The answer comes down to something called basophilia. Look at the pink box at the bottom of this slide. Basophilia comes from the words baso, meaning base, and philia, meaning love or affinity. So basophilic literally means base-loving. Here's the chemistry in plain English. Both DNA and RNA molecules have a backbone made of phosphate groups. These phosphate groups are negatively charged, they're acidic. Hematoxylin and toluidine blue are basic dyes. They carry a positive charge. And what happens when you put a positive charge next to a negative charge? They attract each other, just like the opposite poles of a magnet. So the dye binds to the DNA and RNA in the nucleus, producing that characteristic blue-purple stain. This is exactly what Gray's Anatomy describes when it says these molecules are detected through their acidic components, the phosphate groups. Let's really cement that concept of basophilia, because it comes up again and again in histology exams. Look at these 4 steps on the slide. Step 1: DNA and RNA have phosphate groups on their backbone. These are negatively charged. Step 2: the stains we use, hematoxylin, toluidine blue, are basic dyes. They carry a positive charge. Step 3: positive attracts negative. The dye binds tightly to the phosphate groups through ionic bonding. Step 4: the result? The nucleus stains blue-purple, making it easy to see and identify under the microscope. Gray's Anatomy uses the phrase, DNA and RNA molecules are said to be basophilic because of the binding affinity of their negatively charged phosphate groups to basic dyes such as hematoxylin. That sentence is exam gold. Make sure you understand every word of it. One more thing to note. The text also mentions the Fulgen reaction, and that's what our next slide is all about. Now we come to the Fulgen reaction, described in Gray's Anatomy as a specific stain for DNA. And this is where things get really fascinating, because the story of how this stain was discovered is a brilliant piece of scientific history. The researcher, Robert Fulgen, was a German physician and biochemist born in 1884. He spent years studying nucleic acids, and in 1914, he began developing a method to specifically stain DNA inside cells. His work was so significant that he reported it at the 8th German Physiology Congress in 1923, and in 1924, together with his collaborator Heinrich Rosenbeck, he published the landmark paper, Mikroskopisch chemischer Nachweis einer Nukleinsäure vom Typus der Thymonukleeinsäure. Don't worry about the German title. It translates roughly to microscopic chemical detection of a nucleic acid of the thymonucleic acid type. In other words, they proved you could see DNA inside cells specifically. Fulgen also made another remarkable discovery, that both plant and animal cells contain nuclear DNA. At the time, this wasn't fully established, and his work helped confirm it. How does the Fulgen reaction work? Here's the mechanism in 4 steps. Step 1. Acid hydrolysis. The tissue is treated with hydrochloric acid. This removes the purine bases from the DNA, specifically adenine and guanine. But here's the key. It only works on the deoxyribose sugar of DNA, not on the ribose sugar of RNA. Step 2. Exposed aldehydes. The removal of these bases exposes free aldehyde groups on the deoxyribose sugar chain. Step 3. Schiff's reagent. The tissue is then treated with Schiff's reagent, a Fuchsin-based dye. This reacts specifically with the exposed aldehyde groups. Step 4. Color reaction. The result is a distinctive magenta or pink-purple color, only where DNA is present. And here's the genius of it. RNA doesn't get stained. Why? Because RNA has a 2OH group on its ribose sugar. This structural difference means acid hydrolysis doesn't expose the same aldehyde groups in RNA, so Schiff's reagent has nothing to react with. This is why the Fulgen reaction is called a specific stain for DNA. It distinguishes DNA from RNA, something hematoxylin cannot do, because hematoxylin stains both. In practical terms, this reaction is used in cytology and pathology to measure DNA content in cells, which is hugely important in cancer diagnosis, because cancer cells often have abnormal amounts of DNA. Now this is my favorite slide in the whole presentation, because it brings everything to life. On the left we have figure 1.1, the beautiful color diagram of a typical cell. This is the kind of illustration that's been in textbooks for decades. Everything is labeled clearly, the nucleus in pink, the nucleolus inside it, the nuclear envelope, the double membrane surrounding the nucleus, and the nuclear pores, which are tiny channels that allow molecules to enter and exit the nucleus. Point to figure 1.1 on screen. You can also see the mitochondria, those sausage-shaped purple organelles that produce energy. The rough endoplasmic reticulum studded with ribosomes. The centrioles, important for cell division. The lysosomes, the cell's recycling system. But here's the thing about a diagram. It's an artist's interpretation. It's idealized. Now look to the right, figure 1.2. Point to figure 1.2 on screen. This is a real electron micrograph, specifically a transmission electron micrograph, or TM, taken by Dr. Bart Wagner at the Histopathology Department of Sheffield Teaching Hospitals in the UK. This is an actual ciliated columnar epithelial cell from human nasal mucosa. So this is from the lining of someone's nose, photographed at incredible magnification. Look at the nucleus. It's that large, round, darker region in the center. You can see the nuclear envelope, the double layer of membrane surrounding it. Inside, you can see the nucleolus, labeled N, that darker spot within the nucleus. The label EN refers to the euchromatic nucleus, the lighter staining regions where DNA is in an active, open form and genes are being read. The darker regions are heterochromatin, where DNA is more tightly packed and less active. And look at those mitochondria labeled M. You can actually see their internal folds, called cristae, at this magnification. Compare that to the schematic on the left. The diagram helps you understand. The micrograph shows you reality. This is exactly why Gray's Anatomy includes both, and why you should always study both. Before we move to the quiz, let's quickly consolidate the key terminology, because in your exams, you will be expected to define these precisely. Nucleus, the largest intracellular organelle, 3 to 10 micrometer, spherical or ellipsoid, houses DNA and RNA, controls all cell activities. Basophilic, having affinity for basic dyes due to negatively charged phosphate groups on nucleic acids, DNA and RNA. Hematoxylin, a basic dye that stains DNA and RNA blue-purple by binding to their negatively charged phosphate groups. Most widely used routine histological stain. Fulgen reaction, a staining method specific for DNA. Uses acid hydrolysis to expose aldehyde groups on deoxyribose, then shifts reagent produces a magenta color. DNA, deoxyribonucleic acid, double-stranded molecule carrying genetic instructions, found in the cell nucleus. RNA, ribonucleic acid, single-stranded, helps execute genetic instructions, transcription and translation, found in nucleus and cytoplasm. Learn these definitions. Not just the words, understand the concepts behind them. When you understand why something is called basophilic, you never have to memorize it. It just makes sense. Let's take a moment to appreciate the people whose work we've been discussing, because science doesn't happen in a vacuum. It's built by real human beings, often over decades of painstaking work. Robert Fulgen. Born in Lahr, Germany in 1884, Fulgen studied medicine and became deeply interested in the chemistry of nucleic acids. He developed his famous reaction over years of work beginning around 1914. His 1924 paper with Rosenbeck changed how we study cells forever. He continued to work until his death in 1955. His name lives on in every histology lab in the world that uses his reaction. Heinrich Rosenbeck. Rosenbeck was Fulgen's collaborator on the landmark 1924 paper. The full references, Fulgen R and Rosenbeck H, 1924, Mikroskopische chemische Nachweis einer Nukleinsäure vom Typus der Thymonukleinsäure, published in Hauptsäuler's Zeitschrift für Physiologische Chemie, Volume 135. This is the paper that described the reaction in detail for the first time. Dr. Bart Wagner, Sheffield Teaching Hospitals, UK. The electron micrograph you've been looking at, Figure 1.2, is the work of Dr. Bart Wagner from the Histopathology Department at Sheffield Teaching Hospitals, UK. The image shows a ciliated columnar epithelial cell from human nasal mucosa. The detail you can see in that image represents extraordinary skill in specimen preparation and electron microscopy. Gray's Anatomy credits him specifically, and so do we. Remember, every figure in a textbook like Gray's Anatomy represents someone's expertise, dedication, and scientific career. These aren't just pictures. They're contributions to human knowledge. Okay. Time to test yourself. I'm going to give you the question first. Pause the video if you want to think about it before seeing the answer. Ready? First question, what is the typical size range of the nucleus? Answer, 3 to 10 micrometers in diameter. Don't forget the units. Second question, why are DNA and RNA called basophilic? Answer, because they have negatively charged phosphate groups that attract basic dyes like hematoxylin. The ionic interaction between negative phosphates and positive basic dyes causes the staining. Third question, what color does hematoxylin stain the nucleus, and why? Answer, blue-purple. Because hematoxylin is a basic dye that binds to the acidic phosphate groups of DNA and RNA through ionic bonding. Fourth question, what makes the Fulgen reaction specific for DNA and not RNA? Answer, the Fulgen reaction uses acid hydrolysis to expose aldehyde groups on deoxyribose, the sugar in DNA. RNA uses ribose, which has a 2-OH group that prevents the same reaction. So only DNA produces the magenta color which shifts reagent. Fifth question, who developed the Fulgen reaction, and when was it published? Answer, Robert Fulgen and Heinrich Rosenbeck, published in 1924 in Hauptsäuler's Zeitschrift für Physiologische Chemie. So, how did you do? If you got all 5, brilliant! If not, no worries at all. Go back, rewatch the relevant section, and try again. And that brings us to the end of today's video on the nucleus. Let's do a final 30-second recap. The nucleus is the largest intracellular organelle, 3 to 10 micrometer, spherical or ellipsoid. It contains DNA and RNA, which are basophilic, meaning they attract basic dyes through their negatively charged phosphate groups. Hematoxylin and toluidine blue are conventional stains that color the nucleus blue-purple. The Fulgen reaction is a specific stain for DNA, developed by Robert Fulgen and Heinrich Rosenbeck in 1924, producing a magenta color through acid hydrolysis and Schiff's reagent. If this video helped you, please give it a like. It takes 1 second and it genuinely helps this channel reach more students who need this content. Subscribe to Gray's Gyan so you never miss a new video. We cover anatomy, histology, physiology, and more, all explained clearly step by step. Drop a comment below, tell me which part of the nucleus you found most interesting, or ask any question you have. I read every comment. And share this video with a classmate who's struggling, because that's what learning is about. We rise together. Until next time, keep studying, keep asking questions, and I'll see you in the next video.